Next, we're going to hear from Lee Kugelmann. Lee is a genetic counselor at UF Health's Norman Fixel Institute for Neurological Diseases, and she completed her genetic counseling training at Emory University. Lee currently works at UF's Neuromuscular, ALS, Huntington's, and Movement Disorders Clinic. So thank you very much, Lee, for being here today. I will stop sharing and let you go ahead and present your slides.
Okay, thank you guys so much for having me. Let me get my slides up and make sure. So I just want to make sure you guys are looking at the presentation and not my notes view, correct?
Correct.
Great.
Thanks.
All right. Well, I'm excited to talk to you guys today. Just to start off, I have no conflicts of interest to share. So I'm going to talk through, we're going to start off with an introduction to genetics, just so we're all on the same page with all of the terminology. And then I'm going to talk through the different inheritance patterns for a variety of different neuromuscular diseases. And then I'll get into the weeds on what the genetic testing process can look like. So to start us off, here's some basic biology. So our DNA, that's our collection of our genetic material. And a small chunk of that DNA that has a specific task, that's what we call a gene. Didn't mean to go forward. Our DNA is wound up stored in these structures called chromosomes, and these exist inside every cell in our bodies. So this is a cartoon version, but here's what our chromosomes actually look like under a microscope. You can see that we have one of each. They come in pairs. So we get one copy from our mom and one copy from our dad. And for everybody, all of the first 22 pairs are the same, except for these last two. These are what we call the sex chromosomes. So you might have heard of X and Y chromosomes. This is what determines a person's sex. So this individual here, they have one X chromosome, that's this long one, and then one Y chromosome here. So this person is a male. So I kind of like to think of our genes like a recipe. It tells you the instructions to add all the ingredients, so the flour, butter, sugar, to make something. Only instead of making something like this cake, our bodies are making a protein. These are the proteins that we need to grow and function and do all the things our body needs to do. And so the recipe, the genetic code, is made up of these series of letters. So the body reads through this code and follows the recipe to make these proteins. So for the rest of this presentation, I'm going to represent our genes like this to show the two different copies here, a dark copy and a light copy. So when there's some sort of change in the recipe, so like here the recipe doesn't have any flour, we're not able to follow it. We're not able to make whatever it is we're able to make. So the body can't read those instructions, and then it can't make the protein that it's supposed to be making from that gene. So you probably heard a lot of different words to explain what this is: mutation, variant. I like to use the word gene change, but all of these basically mean the same thing. There's something in the recipe that's missing or changed that isn't allowing the body to read through and produce the protein. So for this presentation, I'm going to note a gene change by this red marker here. So now I'm going to go through a couple different types of gene changes that we see in neuromuscular diseases. So one is a substitution. And so that happens when one letter in the code is replaced with a different letter. So in this example here, the code is supposed to have a G at this spot, but instead there's an A. So that means when the body is reading through, it's going to read something different and not be able to follow this recipe right. Another type of gene change is a deletion, and this is common in Duchenne muscular dystrophy. So that's where we're missing several of the letters in the code, and so the body reads through and it comes to this point where there's nothing there. And again, that means it's not able to do whatever the instructions for this gene, the recipe for this gene, is telling it to do. And then another option is a duplication or a repeat. So this is the cause of disease in conditions like myotonic dystrophy, Friedreich's ataxia, where you can see we have this series of letters, this CGG, and then it gets repeated here, and that causes disease. All right, so now to talk a bit about inheritance patterns. So when we say inheritance patterns, what we mean there is how the disease is passed down through the family tree. And knowing the inheritance pattern for a disease is really important because it can help us as the providers counsel you guys on the recurrence risk. And what we mean by that is who else in the family is at risk to have symptoms of this disease. So to figure out the inheritance pattern, we need to know where and how many gene changes are required for a person to have symptoms. The first one I'm going to talk about is dominant inheritance. So that's where you only need one copy of a gene to have a change in order to have symptoms of a certain disease. So for down here, this person, one copy, their dark copy has this gene change here. The light copy does not. This person would have symptoms of a dominant disease. This person has no gene changes, so they wouldn't have symptoms. So some conditions that might be represented here in the audience that follow a dominant inheritance pattern are Charcot-Marie-Tooth disease, myotonic dystrophy, ALS, but there's lots of others. So I'm going to go through an example here of what passing down a dominant disease can look like. So this is a person here, let's say this is a person with myotonic dystrophy. So they have one copy of their gene chain that has the change or the repeat that causes myotonic dystrophy. The other copy doesn't have a gene change. So when we all go on to have kids, we pass down at random one of our copies. And it's 50-50, completely random, whether we're going to pass down this dark copy or the light copy. Same for their partner, the other parent. It's completely random if they're going to pass down the dark copy or the light copy. So this parent here with myotonic dystrophy, they pass down the copy with this gene change to their children. No matter what these children inherit from the other parent, these children are going to have myotonic dystrophy. But if the parent passes down the light copy, the copy without the gene change, these children are not going to have myotonic dystrophy. So when someone carries a dominant disease, we say there's a 50% chance risk that they're going to have a child who also has that disease. So the next one I want to talk about is recessive inheritance. And so recessive diseases, you need a change on both copies of the gene in order to have symptoms. So this person down here, there's a gene change on the dark copy and a gene change on the light copy. So this person does have symptoms of a recessive disease. If a person just has a change on one copy of a gene, we call them a carrier. So these two people here, this person has a change just on the dark copy, but their light copy does not have a change. This person, the dark copy doesn't have a change, but the light copy does. We call them carriers. Typically, carriers of recessive disease don't have symptoms, but they could have children who have the disease. So some conditions with recessive inheritance, Charcot-Marie-Tooth, again, can also follow recessive inheritance depending on the specific gene. SMA is another example. So we'll go through, let's say that these two people are carriers of SMA. So this is a parent that has one gene change on one copy and nothing on the other. Same with this parent. So when they go to have kids, remember it's 50-50 which copy they're going to pass down. If both parents pass down the copy with the gene change, so this one passes down the dark blue, this one passes down the dark or the light orange, this is a child that will have SMA. But if only one parent passes down a copy with the gene change, so for example, if this child here who got the dark blue copy with the gene change from one parent and the dark orange copy without the change, or this child over here who got the light blue copy without the change and the light orange copy without the change, these two children would be carriers. So they wouldn't have symptoms of SMA, but they would be at risk to have children with SMA. And then, of course, if you get a copy, the copy you get from each of your parents neither has a change, you will not have symptoms, and you won't be at risk for having children with SMA because we can't pass down what we don't have. So when two parents are carriers of a recessive disease, we say there's a 25% chance that they could have a child with that recessive disease. As you can see here, this 1 out of the 4 possible options where they have a child with SMA or another recessive condition. There's a 50% chance that they could have a child who is a carrier. So you can see 2 out of the 4 possible options where they have, they get just 1 copy of the gene change. So then if someone who has a recessive disease, say someone who has SMA, goes on to have children, when they're passing down their genes, no matter which copy they pass down, they're going to pass down a copy with a gene change. Now, in this example, we're assuming that their partner is not a carrier of the same disease. And I'll talk a little bit later about how we can do genetic testing to determine if your partner is or isn't a carrier of disease. But if we're assuming that we're not, an affected parent will have all of their children will be carriers of a recessive disease. So they would be carriers in this example of SMA, but they would not have children with SMA unless their partner is also a carrier. So we say 100% or all children of someone with a recessive disease will be carriers, but they will only have symptoms if their other parent is also a carrier. All right, so the last type of inheritance I'm going to talk about is X-linked inheritance. So that has to do with our sex chromosomes. So you'll remember that typically we say people with 2 X chromosomes are female and people with 1 X and 1 Y chromosome are male. So in X-linked inheritance, we're talking about gene changes that are located on one of the X chromosomes. And your sex determines whether or not you have symptoms of that disease. So some examples of this are Duchenne and Becker muscular dystrophy, which are inherited in an X-linked manner. So here's an example here where a mother is a carrier. Let's say she's a carrier of Becker muscular dystrophy. So one of her X chromosomes has a gene change, the other does not. The father doesn't have any gene changes. He has an X chromosome with no change and a Y chromosome with no change. So the father actually determines the sex of a baby. If the father passes down an X chromosome, we're going to have a daughter. And if the father passes down a Y chromosome, we'll have a son. So if the mother passes down the X chromosome with the gene change, and the father passes down his X chromosome, we get a daughter who is a carrier of Becker muscular dystrophy. If the mother passes down the X chromosome with the change, but the father passes down the Y chromosome, then we have a son who has Becker muscular dystrophy. But if the mother passes down the X chromosome without the change, regardless of what the father passes down, these people will not be carriers, will not be affected. So if a mother is a carrier of an X-linked condition, we say half of her sons, 50% are affected because she either passed down the dark blue, the copy without the change, or the copy with the change. And if a mother is a carrier, we say 50% of her daughters are at risk to be carriers. So for someone who has an X-linked condition, so let's say an example here, this is a father with Becker muscular dystrophy. He's going to either pass down the X chromosome to his daughters or the Y chromosomes to his sons. So if he passes down the Y chromosome, there's no gene changes there. So none of his sons will have Becker muscular dystrophy. But if he passes down the X chromosome, all of his daughters will be carriers. So when someone has an X-linked condition, when a father has an X-linked condition, none of their sons are at risk to have that condition or be carriers, but all of their daughters will be carriers. Okay, so now I want to talk through a little bit about genetic testing. There's a lot of different methods that we use for genetic testing, and targeting the right method, it really depends on what disease does a person have, what kind of symptoms do they have, and what types of gene changes happen in that disease. So we can do sequencing, which is like reading through the genes. If we think someone has myotonic dystrophy or Friedreich's ataxia or another condition that's caused by repeats, we do what's called repeat analysis. Deletion duplication analysis is one type of technology. So for example, if someone, we think they might have Duchenne muscular dystrophy, that's the type of test we would order. Also all sorts of other types of tests. But to kind of speak generally about what the testing process would look like, you would go through a commercial lab. So here's just some examples of some commercial testing labs. Now, some of you may have had genetic testing done at a research lab. So the important thing to note about that, and really the only difference there between a research lab and a commercial lab, is that commercial labs have to follow a law, a federal statute called CLIA, which basically has some rules about how samples are handled and things like that. So if your genetic testing is required for part of your treatment or your medical management, say to get a drug like Spinraza, you need to prove genetic testing for SMA, and you had testing done through a research lab, we may have to repeat testing through a commercial lab that has that CLIA approved standard. Regardless of what lab we do, what test we do, they all kind of follow the same process. The test has to be ordered by a medical provider, your doctor, whoever your care team is. So then you'll collect a sample. There's a lot of different ways we can get a sample for a genetic test. Because our genes are in all the cells in all of our bodies, we can kind of take a sample from anywhere. But the most common ones are either blood or saliva. A lot of times these days we can do saliva testing, and it's just as valid. The analytics are just as strong as doing a blood test. Sometimes for certain tests, like if we're doing repeat analysis, for example, the labs might request that we send a blood test instead of a saliva test, but that doesn't mean a saliva sample is any less valid or strong. Another thing that is an option now is that tests can be done with a home kit. So we can get a kit and have it mailed to your house, and you don't have to come into the clinic to get genetic testing. So when that's all done, the sample gets sent off to the lab, and the lab will go through and look for changes in whatever genes that we recommended when we ordered the test. Then eventually they'll send that report back to your provider, who will share that news with you. So this process of between when you send the sample to when you get the report, it can vary how much time it takes depending on what the test is. I tend to say 2 to 8 weeks, and that's, there's time for the lab to run the test, time for us to deal with working through insurance issues and things like that. Then when we get that back, your genetic counselor, your doctor, your nurse practitioner, whoever's been working with you in getting this test will talk through what are these results, what do they mean for you. So I wanted to talk to you about kind of what some possible results would be. So the important thing then is to take a step back and find out what is the lab doing when they're looking for these gene changes. And so the lab's job is to say, if we find a change in a gene, what does that mean? Because sometimes a change in a gene is completely benign. We all have changes in our genes. That's what makes us all different from one another. So it's the lab job to say, is this a change that changes the way the body reads the gene, or does this not have any impact? So I like to give the example, in English, we can spell the word gray with either an E or an A. So that change doesn't impact how you read this word. But if I substitute it with a different letter, like an X, you wouldn't be able to read this word. So if this was part of the recipe for something you're making, you wouldn't be able to say what comes next and follow the next step in the recipe. So if a change impacts how the body's able to read the recipe, we call that a pathogenic change. And if it doesn't impact, we call it a benign change. So here's all of the possible results that you might see on one of your genetic test reports. So if something is called pathogenic or likely pathogenic, we consider that a positive test result. We found a gene change that causes disease. If we get a result that says benign or likely benign, that means that whatever change we're looking at, it doesn't cause disease. That's a negative test result. Sometimes the lab doesn't have enough information to say, is this a change that causes disease, or is this change that doesn't cause disease? So when that happens, they call it a variant of uncertain significance. So for me, when I get a variant of uncertain significance back, I take a look at what is this gene, what kind of symptoms is this gene associated with, what's the inheritance pattern known for this gene, and then I look at my patient and I see, do those two things match? And if they do, I can talk with the lab about would testing other family members help to clarify the role of this change, to help find out, does this change actually impact someone's disease? So for example, if we find a change in a family member that doesn't have symptoms, well, that might mean that this change isn't causing disease. But if we test another family member who does have the same symptoms as my patient and we find the same gene change, then we might say, oh, this is the picture starting to come together. So at that point, the variant of uncertain significance is really part of a conversation with your providers to see what does this mean for you? Does this fit with your disease, or do we think this might just be kind of noise? And not relevant to your care moving forward. Then I want to go through some of the different types of genetic tests that we can order. So targeted testing is for situations where we know the gene and we know the variant in the family. So for example, if you came in and said, you know, my mom has CMT, I think I might have CMT, here's my mom's test result, can we test for this? And so we would look specifically for that individual gene change. If we don't know what the gene change is, but we know what gene, so for example, if you say, you know, I have a family member with Duchenne, but I don't have a copy of their test report, I don't know exactly what their deletion was, we could do what we call single gene testing, where we just look at the DMD gene. A panel test is one test that looks at multiple related genes at once. We order a panel test if, say, I have someone in my clinic who has muscular dystrophy, but I'm not sure exactly what type of muscular dystrophy, for example. So I look at a bunch of different muscular dystrophies all in one test. And then if these types of tests come back negative, we have someone who has symptoms, we really think that they have a genetic disease, but all of the previous tests has come back negative. We haven't been able to figure out exactly which gene is causing this person's symptoms. We might look into what we call exome sequencing, which is a really big, broad test that looks at all of the coding regions in someone's gene. So these are all unrelated genes that we can look at. It's a big test, and it's got a lot of benefits as well as a lot of limitations. So your doctor will talk through, you know, if that's something that you're considering or something that your care team is considering, they'll have to talk through a lot with you to make sure that exome sequencing is the right test to order. And then there's the option of carrier testing. So carrier testing is used for, you know, for example, if you have a sibling with a recessive disease. So if you say you have a sibling with SMA and you want to know, am I a carrier of SMA? Or you know you're a carrier and you're looking to have children with someone, you want to find out, are they also a carrier of the same condition? That's what we would do there, is what we call carrier testing. So then the question I always get asked is, what are the costs of genetic testing? And unfortunately, this is really tricky to answer because it depends on a lot of things. Each insurance plan is going to have their own policies for what they cover in terms of genetic testing. They may work with different testing labs, and then depending on the tests we ordered, they're all going to cost something different. So part of my job as a genetic counselor is working with my patient and their insurance and trying to figure out what is the right test to send to the right company that is going to be most financially feasible for the family. Some other things to know are that some labs are offered sponsored testing, where they have funding to put forward testing and it's offered at no cost to the patient. Some labs also offer financial assistance. So if you're trying to get some genetic testing done and it's just too expensive right now for you, be sure to ask about a lab's financial assistance program and see if they can work with you on getting a number or cost that is going to be feasible for you. So the last thing I wanted to mention is what my job is as a genetic counselor. So I see people at all stages of their journey with a diagnosis, before, during, after, you know, years after they've had a diagnosis even, because I can do a lot of different things. So I can help before a diagnosis figure out what is the right test to order and where should we order it from. When we get that test back, I talk through what are these test results, what do they mean for you, what do they mean for other people in your family, other people in your life. And then I can talk to you about how do you want to share these results with your family members. So, you know, say you get a test result back that lets you know that maybe your sibling could be a carrier of this disease. How do you want to have that conversation with your sibling and let them know this information, and what should their next steps be? I also check in with people on how are you adapting to a diagnosis? How are things with your life changing? You know, are things changing with your disease? Have you started, you know, a new treatment? Is that working for you? Things like that, because part of my job is also to check in on the well-being of my patients. So in that, I like to always connect people to resources where they can meet with other people with their condition, for example, the MDA. So here's just a few links I wanted to share with you guys of some information or places to get some more information. If you're looking for a genetic counselor in your area, so someone who can help you facilitate getting some genetic testing or talking through what your genetic testing means, you can go to findageneticcounselor.com and put in your zip code and find someone who's close to you. And the MDA has a great fact sheet for genetics if you want to print out or take a look at to serve as a resource. All right, so that is all I have, but I'm happy to take any questions.
Thank you, Lee. If someone had a genetic test that was done 15 years ago that was inconclusive, what would you recommend that they do now?
Yeah, so definitely share that test with your providers.
Okay.
So a lot of times with inconclusive tests, particularly if it's what we call a variant of uncertain significance, the lab is only able to make a call based on what the medical literature says at this time. So when the medical literature eventually updates, we can take another look at that and say, oh, we've learned a lot more, and now we can actually say, what does this test mean?
Okay. Okay. And I guess, something that's been brought up before is if something's inconclusive, would you recommend having that genetic test done yearly to see if they can get to the bottom of that? Or what, how do you, it's such a hard confirmation to read, to get inconclusive because it's not a yes or a no. So,
Yeah.
At what point should someone kind of just take that and know that it's uncertain, or do they keep going back and trying to see what they have?
Yeah. So I think there's kind of a few things there with that question. So one has to do with, do we need to repeat the genetic test itself? And that really depends on how long it's been since the test was done. So if it was 15 years ago and the lab is no longer functioning, things like that, we may need to get a new sample. But if your genetic test was done, 3 to 5 years ago at, let's just say GeneDx, for example,
Mm-hmm.
they'll still have records of your data, your genetic data that they looked at. So we would then be able to call up the lab and say, hey, could you pull up this person's test result and let us know, have there been any updates that changed the way you interpreted this result?
Okay.
So whether we need a new sample really depends on that. How long has it been since the initial genetic testing?
Got it.
And then to answer the other part about, an inconclusive test result, do we leave it as it is inconclusive, or do we pursue more? I think that's going to really vary person to person.
Mm-hmm.
For some people, it may be that, okay, this test is inconclusive. We're not really thinking that we're going to get any more out of it. We need to look at other types of genetic testing to pursue.
Okay.
For other people, a test is inconclusive, but we really don't know what other avenues to go down. So we're just going to need to wait until, as a scientific community, we learn more and understand these diseases better.
Mm-hmm. And we're definitely progressing that way right now.
Absolutely. Yeah, and I'm part of a research project here at UF that's looking to do some further broader testing to discover new genes and work with people who are in that situation. There's a lot of options for people who have kind of exhausted all of the available standard clinical tests, and now need to pursue the discovery of some new genes. So for example, the Undiagnosed Disease Network works with people who have had a whole bunch of clinical genetic testing that hasn't been able to find an answer, and they can try to pursue on a research basis discovering some new answers there.
That's so interesting. Thank you very much, Lee. I don't see anything else that has popped through, so thank you very much.
Okay, you're very welcome.
Have a great Saturday.